Sure. Hi, everyone. Thanks for being here today. My name is Emily May. I'm the Agricultural Conservation Lead with the Xerces Pesticide Program. And I work with farmers and land managers, researchers, and communities around the country to reduce pesticide risks to pollinators and support pollinator conservation across landscapes. So today I'm going to be talking about monarchs, not just their incredible migration or the importance of milkweed, which you all already know, but a little bit more about the less visible threat of pesticide exposure and what we can do to create safer and higher quality habitat for them across all kinds of settings, from farms to backyards. Every year across North America, monarch butterflies make one of the most extraordinary migrations of the insect world. They travel thousands of miles across a patchwork of different landscapes, and it takes multiple generations to complete the journey. In the eastern population, monarchs are moving north in the spring and summer and laying eggs along the way. And then in late summer, a special generation is born, one that isn't reproducing right away, but is instead beginning the long flight to overwinter in Mexico. And then in the Western population, there are milkweed and monarchs breeding in nearly all parts of the West from June to August. And then the butterflies migrate from inland breeding areas to groves along the Pacific coast, and they cluster in these coastal groves of trees to survive the winter. That fall migration is triggered by day length and temperature, and butterflies are moving many miles per day. So let's take a look at the monarch life cycle, starting with the egg stage, lasting about 3 to 5 days. Eggs are laid on milkweed, which is the only host plant, or it's the host plant that monarchs are using. And then next comes the larval stage, the caterpillar, lasting 10 to 14 days. During this time, caterpillars are feeding constantly and molting through multiple instars. Then they move into the chrysalis. This is all, I think, basic information for the folks on this webinar, but I always like to look at sort of the timing of it. The pupal stage lasts several days to a couple of weeks, and then they metamorphose into adults, and the adult butterfly emerges. Most of the breeding generation monarch adults are living 2 to 5 weeks, and then the migratory generation in late summer can live 6 to 9 months, so long enough to travel back over winter and start the cycle again in the spring. This entire cycle from egg to adult can take about a month, and there are multiple generations each year. So monarchs are interacting with the landscapes and its risks and its nectar plants and its host plants all season long. As you all know, monarchs are facing a lot of problems. They're in decline. Eastern monarchs have declined by about nearly 90% since the 1990s. Western populations have dropped even more sharply by more than 95%. And the drivers of this decline include habitat loss, both in the breeding and migratory range and at overwintering sites, as well as climate and pesticide use. So before 1993, the primary driver was forest loss of the eastern population, was forest loss in Mexico, as well as loss of Midwestern prairie habitat. And then declines from the mid 90s to mid 2000s were driven, at least in part, by the increasing use of glyphosate and loss of milkweeds in and around agricultural fields. We lost about 1 billion stems of milkweed as those herbicide resistant crops became the dominant form of production in the Midwest. And then after the early 2000s, climate and then county level pesticide use, especially the use of neonicotinoid insecticides, appeared to be the main drivers of continued declines. All of these challenges that monarchs are facing interact with each other. And so it's important to address each as we try and help the species avoid extinction. But today I want to talk a little bit more about pesticide risks to monarchs, because monarchs are exposed to very complex mixtures of pesticides across their life cycle and often at every life stage. Pesticide residues show up everywhere we look for them, whether that's in ag fields, in backyards, and wetlands, in the Arctic, in public lands. So monarchs aren't exposed to a single product or single dose. They're exposed to these complex blends. And these mixtures have unpredictable effects in combination. Pesticide use across the landscape is very widespread. This is a map of agricultural use of just one chemical, clothianidin, which is a neonicotinoid insecticide commonly used as a coating on seeds of row crops. Again, this is just one insecticide, but there's many different pesticides that are applied across hundreds of millions of acres and different landscapes, not just agriculture. Over my lifetime, the overall volume or the amount of insecticides that have been applied to crops in the US has gone down, but the toxicity to insects, at least as measured by toxicity to honeybees, has more than tripled over that same time period. And this range-wide use of pesticides poses a challenge to building safer spaces for monarchs. One key thing that we all need to understand is the pathways by which monarchs and other insects can be exposed to those pesticides when they're used. Adult monarchs are flying around, sipping on nectar from flowers and laying eggs on host plants. When they're moving through an area, they could get sprayed directly or pick up contaminated nectar from a previous application. But adults have that benefit of being very mobile. Larvae, on the other hand, can't move out of a contaminated area. So they might pick up residues on the leaf surfaces of host plants or in the actual leaf tissue. Some pesticides, including things like the neonics, are systemic, meaning they dissolve readily in water and they can be taken up into a plant, into its leaves or its pollen and nectar. So because these larvae are essentially kind of stuck in place until they've finished development, they can be chronically exposed to pesticides in and on the leaves of milkweed that they're living on or feeding on. And so for this reason, we really want to make sure that breeding habitat and host plants are adequately protected from that pesticide risk. To make sure that you and I are using the same language, the word risk has a kind of specific meaning when it comes to pesticides. There are two main things that factor into how risky a pesticide is, whether it's to a human or your pet or a butterfly. The first being toxicity. So how toxic is it? The capacity of that pesticide to cause harm. In what way? Does it work inside the body, outside the body? So the other piece is exposure. So how much of the pesticide is that pollinator or person or pet exposed to? Was it exposed on the outside of its body, or did it eat it in the leaves of its host plant, for example? And then how long was it exposed? So all of this really boils down to the dose makes the poison. A highly toxic pesticide that a pollinator is minimally exposed to might be less risky than a less toxic one that they're constantly exposed to. And of course, the most risky would be a high dose exposure with a highly toxic pesticide. So this equation is kind of helpful for thinking about what are the ways we could limit risk, including seeking out different ways to manage pests or otherwise limiting either the harm or the exposure. But what do we know about how toxic a pesticide is? Before a pesticide can be registered with the Environmental Protection Agency, pesticide manufacturers are required to submit the results of a variety of different tests to EPA, looking at toxicity to birds, mammals, fish, and insects, depending on sort of the expected use patterns of that pesticide. And the way that EPA then assesses how toxic it is to insects is based on sort of standard lab testing of adult honeybees, which are the model organism for all other terrestrial insects. So in that test, a pesticide is applied at different doses. The standard test is applying it to the top of the thorax of an adult honeybee, like in this picture. And they then look for the dose that kills half of the tested group of honeybees over the next 48 hours. The smaller the dose, the more toxic that pesticide is to bees. And that sort of standard dose is called the lethal dose, or the LD50. There are many pesticides that have toxicity to bees and other beneficial insects like monarchs that isn't captured by this particular standard test. Things like toxicity to larval insects, native bees, and other totally different groups of insects like butterflies, they may be more or less sensitive than adult honeybees to different kinds of pesticides. Only a very few number of chemicals have been tested specifically on monarchs. And so from that limited toxicity data that we do have, it appears that monarch butterflies tend to be less susceptible than the honeybee to the widely used neonicotinoid insecticides. but they are very, very susceptible to chlorantraniliprole, which is a diamide insecticide that has more than 100 uses across both agriculture and residential settings, including as increasingly a seed treatment on row crops. And it's also now the most popular lawn grub insecticide. It's considered lower risk for bees, but it's very high risk for monarchs. And then pyrethroid insecticides, which are the most common backyard mosquito treatment, are also very highly toxic to monarchs. Many important effects of pesticides are also missed when toxicity tests only focus on adults and on that sort of short-term 2-day window. So for example, when studying the impacts of pesticides on monarch caterpillars, University of Maryland professor Niranjana Krishnan identified this important secondary mode of action, which is the way a pesticide works, a mechanism for harm, for neonics and for some other pesticides, which is that they can disrupt molting at the final stage of larval development. So that's what you see in this picture. It's called arrested pupal ecdysis, which is where the larvae basically can't successfully pupate. They get stuck. And this was in caterpillars that had been exposed to a neonicotinoid, and there were no other symptoms observed prior to their death at this stage. But this was the leading cause of death for fifth instar larvae treated with imidacloprid. And so this is why we need more testing that looks at longer term exposure and at multiple life stages of these insects. It can also, pesticide exposure can have a variety of other harmful side effects that might not kill an insect in 2 days, but can affect their ability to survive and reproduce over the long term. Some of these things are hard to characterize, especially in a migratory species like the monarch, like carryover effects of pesticide exposure from one generation to the next. So to date, we really don't have pesticide regulations that address many of these kinds of harms. Some of the examples might include reduced body size, reduced larval growth or slower growth, smaller wings, shorter lives. So as one example, in a monarch study, they found that monarchs exposed as larvae to fungicides had smaller wings as adults, and that could affect their ability to migrate. That part hasn't been tested, but it suggests that they could have more trouble migrating. So yeah, these are all very important also side effects that you see from different doses of pesticides that monarchs are exposed to. So the broader point I'm trying to make here is that pesticide risk is often underestimated, including in the sort of regulatory process that allows pesticides to be registered and allowed for sale on the market. EPA's assessment tools are often falling short of capturing these risks to monarchs. What's required for registration simply doesn't provide enough information to assure that these pesticides are safe for monarchs, right? There's very limited toxicity data for butterflies. And importantly, EPA is only looking one chemical at a time when they're doing their registrations. But monarchs aren't just exposed to one chemical one time. They're exposed to mixes, overlapping applications, residues in soil, vegetation. Oh, I guess monarchs might not be exposed to soil, but in plants in different parts of their life cycle, combinations of different pesticides, insecticides, fungicides, herbicides, et cetera. But the risk is really only assessed, one chemical at a time. So what ultimately this means is that following a pesticide label is not going to be enough to protect monarchs. So obviously we want everyone to be reading and following labels very carefully, but we also need to do more than that to be able to protect monarchs and other species like them from pesticide risks. So let's talk about habitat a bit and how it gets contaminated. Pesticides don't stay, unfortunately, where they are applied, and plants around agricultural fields commonly contain pesticide residues, including both from things like foliar sprays, as well as from seed coatings of pesticides on seeds. The takeaway that I have from lots of different field trials looking at pesticide residues is that I wouldn't want my pollinator habitat downwind of aerial insecticide applications or like right next to tractor mounted applications. Having even a small buffer between the crop and your habitat can be useful to make sure that you're not getting that highest rate of drift deposition in your habitat. But the other takeaway is that pesticide drift and contamination is not just an agriculture problem. Xerces conducted a study looking at residues in milkweeds across California's Central Valley. So we sampled 227 milkweed plants from a mix of different landscape types, agriculture, urban, natural areas, and every single plant from every single kind of landscape contained pesticides, some at levels known to be harmful to monarchs. Across those samples, there were 25 insecticides, 27 fungicides, 11 herbicides, and a spray additive with an average of 9 pesticides per plant. Again, EPA is looking at these one at a time, but it is really the rule and not the exception for a monarch in this kind of heavy use environment to be exposed to many different pesticides over its lifetime. We conducted a similar study of milkweeds coming out of retail nurseries and found similarly widespread contamination. But one big limitation is that we have very little monarch toxicity data to be able to really understand what the implications of these residues are for those insects. One other thing we really learned from these studies is that many studies are only looking at a small number of compounds, often the ones they're expecting to find, like a neonicotinoid. But the reason we detected so many pesticides is that we were testing for a broad panel of pesticides. We just published a study this past year looking at butterfly host plants in Sacramento and Albuquerque, and we're surprised to find pretty widespread contamination with atrazine in urban areas. Atrazine is an agricultural herbicide. So if we were only testing for pesticides we were expecting to find in urban areas, we wouldn't have seen it. So we really need more sampling, and we need to be testing for a wider range of chemicals, not just the ones that we think we'll find. So yeah, pesticide contamination, just to continue the thread, isn't just an agricultural problem. And it's not just breeding habitat that we do need to protect. Overwintering sites are particularly vulnerable. They're housing hundreds to thousands of monarchs at a time through the winter months. And this is a disturbing video from Pacific Grove, California, which is home to one of the largest Western monarch overwintering sites. In January 2 years ago, volunteers that were doing counts at the site discovered around 200 monarchs exhibiting these kinds of symptoms, dead and dying on the lawn outside of the sanctuary. This was about 1 in 10 of the monarchs that were at the site. And they were showing clear signs of pesticide poisoning. So some individuals were collected, 10 monarchs were collected and sent off for testing, and they contained about 7 pesticides per insect with 3 pyrethroid insecticides at or near a lethal dose in most of these butterflies. So these findings really pointed to pesticide exposure as the likely cause of this monarch kill. While we couldn't pinpoint an actual source of the pesticide contamination at that site, which is surrounded by private homes and a couple of businesses, pyrethroids are really commonly applied in urban settings and in residential settings, frequently used as outdoor treatments for household pests like ants on a patio. They are also commonly used as the sort of barrier treatment, mosquito sprays. If you get your local pest control service coming by every couple of weeks, they're probably applying a pyrethroid like this one, and they can be directly applied to soil, turf, foundations, patios, driveways, ornamental vegetation, flowering plants. So despite California's very detailed pesticide use reporting system, most homeowner applications and many of these landscape treatments go unreported, or they're only reported at the county level. So it makes it very difficult to trace this kind of an incident. So what does this mean for conservation? It means pesticide exposure threatens the conservation success, or the success of conservation work for this species. And it also means that pesticide labels are not protective enough. So in order to recover monarchs, we really have to go beyond these labels to reduce pesticide risk across different landscapes in our fields and habitat and backyards, and through policy and regulation that improves the assessment for this kind of an imperiled species. all this to say, we know pesticides pose challenges for monarchs. And given some of these gaps in the data that we know, it could be worse than we think. So wherever we're looking, we're finding pesticides, and we need to be talking about it, right? Hard conversations are necessary in some of our line of work. Not every space may be suitable for habitat. But on the other hand, we also believe that more habitat for monarchs is good. So we want to spend our time and effort building and prioritizing high quality habitat in safer areas. So let's talk about the solutions. There's many ways to help monarchs and other pollinators, from restoring and protecting breeding and migratory habitat to restoring and protecting overwintering habitat. I know you don't have that in Minnesota, but you got lots of breeding habitat. Reducing risk from pesticide use that you can control and in your community, and then participating in community science and advocacy and policy efforts to help support monarchs and other pollinators. So what makes high quality monarch habitat? There's really three core components, all of which you see here. Native milkweed, the larval food plant and adult energy source for monarchs. Nectar plants, the adult energy source. And then safe habitat, protection from pesticides and high levels of pathogens. Monarchs are nectar generalists, meaning they're going to use a wide range of flowers, but there are certain kinds of species that seem especially attractive or beneficial. So high quality is really just the space where monarchs can thrive safely across every stage of their life cycle. So you already know about milkweed, but depending on where you're located, nectar availability in the beginning and end of migration can be quite important. And so the way that you achieve season-long bloom is planting a variety of different plants with different bloom times. And we have suggested monarch nectar plant lists for different regions of the country available at xerces.org. So check those out. Especially in hotter places, trees and shrubs can be important for providing shade for monarchs, for perching and resting. And there's some evidence that monarchs actually like to lay their eggs on milkweed that's partially shaded, especially in places like California. I'm not so sure whether that's the case also in Minnesota. But so you might be picturing a milkweed planting in full sun, but in hotter areas, we could consider creating monarch habitat that receives some level of shade. There's lots of different opportunities for creating monarch habitat. You saw some amazing pictures from the last presentation about great looking habitat in all kinds of different urban, residential, municipal spaces. We have many different places where we can get even a few more stems of milkweed in the ground. But how do we protect that habitat that we create from the pesticides concerns that I've spent so much time talking about? So if you're starting from scratch, you don't have habitat in the ground already, the starting point is picking a location that's more protected based on what's happening around it. This slide shows two of our pollinator habitat assessment guides. One is for farmland, the other one is for residential areas, for yards, gardens, and parks. And these are really designed to help land managers or gardeners identify what the habitat needs are at a site, including assessing management practices. And each one has kind of a helpful checklist of different practices, things like how are pests being controlled, whether pesticides are used. They're not really a full risk assessment, but they can help you start asking the right questions. And this is, also an opportunity to think about pest control practices if you're bordering a place that you don't control, or if it's in a location where you're in a position maybe to influence management, whether it's at a school, whether it's at a library, or on your own land. So could some of those practices that are used to manage pests be shifted towards non-chemical methods first? Things like mowing, mulching, or physical suppression or removal could reduce reliance on pesticides and help keep that habitat safer for pollinators. Of course, in many cases, you might not have full control over how the land around your habitat is being managed. So then the question becomes, what if your neighbor or even your town is applying pesticides nearby? And this is especially common in residential areas where pesticides are often being applied in ways that are not totally obvious, like they might be if you're next to a farm. We're not just talking about visible sprays, we're talking about things like granular lawn applications, foundation sprays, drenches for things like carpenter ants, termites, and then products that are applied through misting or irrigation systems. Those kinds of applications can still move in ways that unintentionally expose nearby habitat, even when you're doing everything right on your own property. So, what can you do when you don't have control over neighboring practices? What we often look to do is to protect habitat with buffers and vegetative barriers. So, can we physically site habitat in an area that's farther away from where pesticides are known to be used? The farther you can get, the more of a buffer you can provide, the better. Um, and then, in areas that are more space limited, vegetative buffers could be a helpful tool. This is things like basically the same as you would plant a snow fence, right? You can plant something like a conifer fence to help protect from drift. These plantings ideally wouldn't be something that are attracting a pollinator, but something ideal, would be a coniferous, dense species that's capturing that drift before it lands on great, diverse native pollinator habitat. So there's no sort of perfect solution here, but some of these can significantly reduce drift and pesticide exposure in habitat. And then finally, and I know my colleague Sharon presented on this yesterday, but if you're sourcing your habitat plants from nurseries, seek out those providing pollinator safe plants, which could mean buying certified organic plants or simply having a conversation with the person growing your plants to see how they're growing them, and ask them for milkweeds and nectar plants free of pesticides that could be harmful to monarchs and other pollinators. The more that nursery managers are hearing this from their customers, the more that they're going to pay attention to it. So we have a lot more information on that on our website, or you can go back and watch Sharon's talk from yesterday. So those are some of the key steps for habitat protection, siting away from pesticides or changing practices to reduce pesticide risk. Outside of habitat, another way to be engaged in monarch conservation is with community science, which is a really key source of data on how monarchs and their habitat are doing. So if you're in the US, you can participate in a variety of different community science efforts, many of which are on this slide. There's the Monarch Blitz in midsummer, where last summer over 9,000 volunteers contributed more than 19,000 observations, including over 71,000 milkweed plants and 33,000 monarchs. Look up the Journey North, the Monarch Larva Monitoring Project, as well as if you're on the West Coast, Western Monarch Count and the Western Monarch Milkweed Mapper. So all these different programs work together and help people collect and submit data that then helps understand these monarch populations and their distribution. A lot of the work that we do focuses on protecting habitat at a really small scale, one planting at a time. And those efforts are really important, right? They add up. But when it comes to recovering monarch populations at the broader scale, we do have to zoom out. Monarchs don't stay in one place. They're moving across the landscape, interacting with all different kinds of habitats, many of which may be contaminated. So to truly protect them, in order, we need to do what we can at the site level. That is really important. And then realistically, we need to work towards stronger policy, more investment, more effective regulatory frameworks. So one place to start locally is to just start asking questions about how public lands around you are being managed. That includes parks and schools and city landscaping. And then ask for more information about, what pesticides are being used, how often they're being used, and why. Some communities have already adopted pesticide-free policies or IPM plans for their municipal community, or they've restricted the use of certain high-risk chemicals. So that's one place to start in terms of advocacy and policy reform, is to go to your city or your county and advocate for change, like creating an effective integrated pest management plan that prioritizes prevention and non-chemical methods. I would recommend we have a toolkit on this at the Bee City USA website that has a lot of information about these municipal IPM plans. Policy change, really comes about through public advocacy. It's key to pushing these kinds of changes. So whether it's at your school or in your city or your town or your state, your voice really matters in helping policymakers, decision makers come to understand how they can push forward changes in policy that really help protect monarchs and other pollinators. So I think I've pushed through this pretty quickly. But I wanted to point you towards the variety of resources that we have available for free on our website at xerces.org. We have nectar guides available, including information on species that are native and attractive to monarchs for your region, as well as being commercially available. And then some of these other resources that are around protecting habitat from pesticide contamination and sourcing plants that are free from harmful pesticide residues. So with that, I will say many thanks to the Xerces Society members that make our work possible, as well as our other funders and supporters. And I will end my presentation there. Thank you so much for participating in this presentation, and I hope you learned something. And yeah, thanks for having me.